Bismillahirrahmanirrahim. Assalamu alaikum. Shatin Sawati is here and welcome to Biosciences with Shatin Sawati. Welcome to all those who subscribed my channel and welcome to all those who did not subscribe my channel and still watching my channel. And I have a very humble request to all of you to subscribe my channel, promote my channel, share my videos, like my video. If you're sharing my videos, my lecture, you're sharing the knowledge. So keep watching, keep sharing knowledge with other. Today I'm going to start a lecture from my cell biology series, and almost 4 lectures I have delivered about the structure, cell theory, cell membrane, and then cell membrane structure, function, biochemistry, each and everything. Today I'm going to deliver a very important lecture about the most important organelle of the cell, and that is mitochondria. You all are familiar with mitochondria, but today I'm going to tell you something very different about the mitochondria. When I was a student, I was so much confused about the F particles and which are present on the cristae of the in the mitochondrial membrane. I had lots of questions at that time. So today I am going to clear all the questions of all the students related to mitochondria. This lecture is for all students from all the categories. So must watch this lecture. This is very interesting and you will know why the mitochondria is so strong and independent organelle. So I'm going to start my lecture. First of all, I will overview about the discovery of the mitochondria. Mitochondria, often referred to as the powerhouse of the cell, were first discovered in 1857 by the physiologist Albert von Kolker and later coined bioblast means life germs by another scientist Altman in 1886. The organelles were then renamed mitochondria by Carl Blanda 12 years later. Now the question is, when did mitochondria first appear? This is about 1.4 billion years ago. Mitochondria arose through a fateful endosymbiosis more than 1.45 billion years ago. That was suggested by the scientist, that the mitochondria appear first time by the endosymbiosis. And what is endosymbiosis? I will give you a little bit information about the endosymbiosis theory. Mitochondrial ribosomes and transfer RNA molecules and DNA are similar to those of the bacteria. That's why the scientists believe that the mitochondria arised by the endosymbiosis. In that mechanism, what will happen? You can see here aerobic bacteria. This is aerobic bacteria because a mitochondrial structure, like the presence of the ribosomes and transfer RNA and DNA, is more similar to the bacteria. So that's why scientists believe that the eukaryotic cell is created by engulfing the aerobic bacteria and the cyanobacteria. I will discuss it later on about the cyanobacteria, how the chloroplast developed. So here I am giving you just information about the mitochondria. So that mitochondria first was the aerobic bacteria that developed later on into the mitochondria by the endosymbiosis theory. So this was led by the Dr. Lan Margulis in the 1970s to propose an extracellular origin for mitochondria. Is it clear? About the endosymbiosis theory. I will discuss in detail about the endosymbiosis theory, but here I am giving you just an overview that what is mitochondria and how it was developed, and what is the theory, and what the scientists thought at that time, how it developed. Okay. Number of mitochondria. Now, this is very important. Each cell contains a different number of mitochondria, and the number of mitochondria depend upon the function of the cell. That how much energy the cell require. The more energy a cell needs, the more mitochondria that will be present. Cells have the ability to produce more mitochondria as needed. The most important thing you should know about that, the erythrocytes mean the red blood cells are devoid of mitochondria nucleus to accommodate a large amount of hemoglobin. The reason is that why the erythrocytes lack mitochondria and nucleus and so on, to accommodate large amount of hemoglobin, and that hemoglobin, you know that very important pigment which is present in the red blood cells to carry the oxygen. So to accommodate more oxygen, that's why the mitochondria and nucleus absent in the erythrocytes. Okay, now function. Today, mighty mitochondria are known to play a critical role in providing energy to the cell, maintaining, most important, because the mitochondria is a powerhouse of the cell. So providing energy to the cell, maintaining cellular metabolism, as well as regulating cell survival and death. This is very important thing. This help in the cell survival and death. These organelles are responsible for converting food or molecules and oxygen into adenosine triphosphate, means the ATP, energy storing molecule. ATP can then be used to provide energy for the cell. Okay, the concept clear about the discovery and the function of the mitochondria and how the mitochondria developed. What was the view of the scientist about the development of the mitochondria? Okay, now dysfunction of the mitochondria. Not surprisingly, because of their central role in the cell, dysfunction in mitochondria have been linked to numerous diseases. Most recently, defects in mitochondria have been linked to the process of neurodegeneration. That is very important. The progressive death or loss in the function of the neurons, the cells that make up our brain and nervous system. Neurodegeneration, the mechanism in which the neurons degenerate, destroy. So mitochondria play important role in the progressive death or loss in function of neuron. Such damage to distinct subsets of the neuron is the root cause of diseases such as Alzheimer, Parkinson, Huntington's, and amyotrophic lateral sclerosis. All these neurodegenerative diseases occur because of the dysfunction of the mitochondria. Is it clear? Now, the structure and biochemistry of mitochondria. That is very interesting. This is the overall structure. You can see this is the mitochondrial outer membrane. This is the outer envelope, outer membrane. And inside the outer membrane, you can see here, this is the inner membrane. And between the outer and inner membrane, there is an intermembrane space. Must keep it in your mind. The mitochondria is the double membrane bounded organelle. So it has outer membrane as well as inner membrane. So between the outer and inner, there is an intermembrane space. Now you can see the inner membrane is folding in the form of the finger-like projections. And these finger-like projections are known as cristae. One is known as crista and the sum of all known as cristae. Singular crista, plural cristae. Now, the most important thing about the ribosome is that, about the mitochondria is that mitochondria has its own DNA and ribosome. This space is known as matrix, which is a matrix is a fluid. This is the cytoplasmic like fluid which is enclosed in the inner membrane. So you can see here the cristae, you can see the ribosomes. These ribosomes and the presence of the DNA is responsible for the mitochondria to be independent and strong. The reason is that because of the DNA, presence of the DNA and ribosome, the mitochondria has its own machinery for protein synthesis, as well as it is known as the self-replicating organelle because it has its own DNA and it can increase the number of the mitochondria by replication in the cell. So that's why this is so strong and independent, because it do not depend completely on the nuclear DNA. It has its own DNA, it has its own ribosome which help in the self-replication process. So this is all about the outer membrane, inner membrane, cristae, and ribosomes and DNA and matrix. Matrix is like the cytoplasmic fluid which is enclosed in the inner membrane. Now, matrix. The mitochondrial matrix is the site of the tricarboxylic acid cycle. Tricarboxylic acid cycle is also known as the citric acid cycle. That is also known as, you are familiar with that, Krebs cycle. I will discuss in my next lecture about the Krebs cycle and many more. A series of enzymatic reaction initiated by the conversion of the pyruvate and fatty acid to acetyl coenzyme A. Now this pyruvate, you know that fatty acids and the pyruvate is the pyruvic acid which is produced in the cytoplasm by the breakdown of the glucose during the process of glycolysis. So this pyruvate, after producing in the cytoplasm by the mechanism glycolysis enter into the mitochondria through the mitochondrial membrane, and then in the matrix, then it will continue the mechanism which is known as the Krebs cycle or tricarboxylic acid cycle. Pyruvate and fatty acids are transported into the mitochondria from the cytoplasm by membrane-bounded permeases. Now, how the pyruvate and fatty acids, these are the large size molecule as compared to the other ions and proteins. So these fatty acid and pyruvate enter into the matrix of the mitochondria through the permeases. Permeases are the enzymes which increases the permeability of the membrane of the mitochondria, and in this way what will happen, the pyruvate and fatty acid which are present in the cytoplasm, which are synthesized in the cytoplasm, then move or enter into the matrix for the mechanism for continuing the mechanism of tricarboxylic acid cycle, which is known as Krebs cycle. Is it clear? Okay, mitochondrial membrane. Mitochondria are membrane-bounded organelle, but they are membrane-bound with two different membranes. That is very important. They have two different. You can see here, the outer membrane is smooth. This is the outer membrane of the mitochondria which is smooth, and this is the inner membrane which is enfolding. So this character is different from other organelle. That's why this is known as double membrane organelle, and you can say two different membranes. And that's quite unusual for an intercellular organelle. This is unusual for the intercellular organelle. Those membranes function in the purpose of mitochondria, which is essentially to produce energy, contain respiratory proteins, and help in the transport of the proteins into the mitochondria. Not only the proteins, but pyruvate and fatty acids, and many more. The inner mitochondrial membrane is the active site. Now, outer membrane help in the transport of the material from the outer side of the organelle, means from the cytoplasm to the inner matrix. But the inner mitochondrial membrane play a very important role in what? In ATP production. In what? ATP production. Okay. Now you can see here again, this is the structure of the mitochondria, and this is the membrane. This is the inner membrane, and this is the, you can see here, outer membrane. You can see here porins. Porins mean pores. And these pores permeability is increased by the permeases enzymes. So that's why pyruvate and fatty acids enter from the cytoplasm into the matrix of the cell. So you can see here, this is the inner membrane. That is very important and active site for ATP production. ATP produced here, because all the important proteins, I will show you that all the important proteins, enzymes present here. We all are familiar that the membrane is the lipid bilayer, and proteins are also present, which are in the form of the integral protein and peripheral protein and many more. So you can see here the important enzyme like ATP synthase. This is the very important protein, ATP synthase, which help in the ATP synthesis. You can see here the keratin kinase help in the transfer of the phosphate from the ATP for the production of the keratin, and keratin is the most important protein. Now nucleotide kinases, where the word kinase is used, kinase actually used for the transferring of the phosphate from the high energy molecule like ATP for the formation of the nucleotides, keratin, and many more. So nucleotide kinases help in the formation of the nucleotide by the transferring of the phosphate from the ATP to the polynucleotide chain. So this is the overall view of the, which I will discuss later on in transport chain, which occur along the inner membrane. So this is the little bit information about the structure of the cell membrane of the mitochondria. Is it clear to all of you? Now, mitochondrial membrane phospholipids. As we know that the membranes are made up of the phospholipids and proteins. So mitochondrial membrane phospholipids are essential for the mitochondrial architecture. The major classes of the phospholipid found in the mitochondrial membrane are similar to other membranes, such as phosphatidylcholine. I have discussed in the cell membrane lecture in cell membrane biochemistry that phosphatidylcholine, phosphatidylethanolamine, and some are exclusively components of the mitochondrial membrane, such as cardiolipin. That is very important, cardiolipin. I am here giving you some information about the cardiolipin, because most of the students don't know about the cardiolipin. What is cardiolipin? Most of them ask me, what is cardiolipin? So cardiolipin, I am giving you some information about the cardiolipin. Cardiolipin are actually what? They are the lipids, types of the phospholipids. So they are also known as Kolkata antigen, is an important component of the inner mitochondrial membrane, where it constitutes about 20% of the total lipid composition. Total lipid composition, among the total lipid composition, 20% is the cardiolipin. Cardiolipin play an important role in regulating various kinds of the mitochondrial proteins, such as electron transport complex, carrier proteins, and phosphate kinases, which I have shown you that keratin and nucleotide kinases, and is also essential for the organization of the particular mitochondrial structure, such as cristae and the contact sites. Okay. Unlike plasma membrane, mitochondrial membrane contain high level of the cardiolipin, 15% of the total phospholipid, and low level of the sphingolipids and cholesterol. Sphingomyelin and cholesterol are in low level, are low quantity, but cardiolipin is in high quantity. And you are now familiar with the function of the cardiolipin. What is basically the function of cardiolipin? Okay. Now, very important thing about the structure of the inner membrane. Inner membrane, which is enfolding in the form of the cristae, and on the surface of the cristae, there are small particles present. You can see here small projections, small particles. These particles are known as F particles. And this F particle is known as ATP synthase. This is known as ATP synthase. This is present. F particles located within the thylakoid membrane and the inner mitochondria. I will discuss it later on in thylakoid membrane, which is the membrane of the chloroplast. So here I am discussing about the mitochondria. So I will discuss about the mitochondrial F particles. So F particles are known as, these are the small proteins which are present on the projections, which are cristae, and these are known as ATP synthase, mean help in the formation of ATP. Okay. Now this ATP synthase, you can see here the structure of the ATP synthase. This ATP synthase consists of the two parts, F1 part and F0 part. Now, what is F1 and what is F2? The F1 fraction derives its name from the term fraction, F from the fraction 1, and F0 not for used for 0. Now, why it is called F0 and why it is called F1, I am giving you here in detail. So, derive its name, F0 derive its name from being the binding fraction for oligomycin. Now, what is oligomycin? Oligomycin is the naturally occurring, you can say, antibiotic. So, this oligomycin attached to the F0 and inhibit the function of F particles. And F particle function is ATP synthesis, because this is F particle is also known as ATP synthase. So if there is no need of ATP, then oligomycin come and attach with the F0 and stop the activity of the F particle. So that is very important. Must keep it in your mind. What is or what are F particles and what is the other name of the F particle? They are known as ATP synthase, because most of the students are confused about the ATP synthase and F particles. So now the concept clear upon you people that the F particles are known as ATP synthase, and this ATP synthase or F particles consist of two parts, F1 and F0, and the F0 is that part which inhibit the function of the F particle by the attachment of the oligomycin. Oligomycin act like the inhibitor. When there is no need for ATP synthesis, then the oligomycin come and attach with the F0 and stop the activity of the F1 fraction, F1 particle. Okay. F1, F0 particles, you can say that like these are the fractions, these are the subunits of the F particles. Is it clear now? These are the subunits, F1 subunit and F0 subunit, sorry. Okay. Now, the F1, you can see here, You can clearly see here, this is the F1 portion and this is the F0. Is it clear? F1 and F0. And what will happen? This is the F0 at which the oligomycin come and stop the activity of the F1 to synthesize ATP. So this is the base, this is the pedicellar stalk, and this is the head. And now the F1 portion of the ATP synthase is hydrophilic and responsible for hydrolyzing ATP. Is it clear now? And F0 is what? Is a water insoluble protein with 8 subunits and a transmembrane ring. So now the concept about the F particles ATP synthesis is clear. The function of both F0 and F1 particle, these are the parts, subunits of the F particle. Is it clear? I hope so. You are enjoying and the concept is clear upon you people about the ATP synthase F particles. Okay, that is very important discovery. So you can see here the two scientists, Paul Bohr, and the second one is the John Walker. Now, the Paul developed in 1960 to 1970 developed the binding changes or flip-flop mechanism theory, which postulated that ATP synthesis is dependent on the conformation changes in ATP synthase. And the other one, Walker, crystallized the F1 catalytic domain of ATP synthase. And his Paul and his Walker, and both got the Nobel Prize in 1997 for this important information, for this important discovery. Okay. Now this is the inner membrane of the mitochondria, and you can see a number of the cytochromes and ATP synthase, which is known as F particle. And you can see here, this is the ATP synthase at which the ADP is converted into ATP, mean ATP formation occur here when the protons pump from the outer to the inner side of the mitochondria. I will discuss in detail about the electron transport chain, and you can see here the different cytochromes. These are also, and the Q proteins, ubiquinone proteins, and many more along the inner membrane of the mitochondria, which play a very important role in ATP synthesis. I will discuss this diagram in detail in my next lecture. Okay, so you can see this is the matrix and many succinate, fumarate. These all synthesized during the Krebs cycle inside the matrix. And this is the overall view of the mitochondrial inner membrane that play a very important role in ATP synthesis. Okay, this is the electron transport chain. This is another diagram which is showing ATP synthesis along the inner mitochondrial membrane. Okay, I hope so the lecture will be clear for you people, and if you have any question, write in the comment box. So like my video, share my video. Thank you very much. I will be back tomorrow. Allah Hafiz.